operons, and genetic engineering. Designed for listening while driving, includes summaries, quizzes. And Part 1, Gene Expression, Transcription, Translation, and Operons. Section 1, DNA, Proteins, and the importance of folding DNA is the blueprint. Proteins are the machines. In cells, over 80% of resources are dedicated to making proteins. This is because proteins do almost everything. They carry signals, build structures, and catalyze reactions. But these proteins are only useful if they fold correctly. A misfolded protein is like a crumpled instruction manual, unreadable and often harmful. Section 2, From DNA to Protein, the two-step process. Step 1, Transcription. RNA polymerase binds to a promoter, a specific spot on DNA. It reads the gene and creates messenger RNA, mRNA, a temporary copy of instructions. Step 2, Translation. Ribosomes grab onto mRNA and read it in sets of 3 bases, called codons. Each codon tells the ribosome which amino acid to add. The chain of amino acids folds into a protein. Fun fact, in bacteria, mRNA only lasts a few minutes. It's like a fast food order, made, used, and discarded quickly. Mini Quiz 1, Transcription and Translation. Q1, what enzyme is responsible for creating RNA from DNA? A, RNA polymerase. Q2, where does translation occur? Ribosome. 3, what are the 3 base units called on mRNA? Codons. Section 3, Ribosomes in prokaryotes versus eukaryotes. Ribosomes are universal, but slightly different in structure. In prokaryotes, ribosomes are smaller, 70S spare. In eukaryotes, they're larger, 80S. Ribosomes consist of ribosomal RNA, rRNA, and proteins. They attach to mRNA and decode it to build proteins. Section 4, Operons, efficient gene control. An operon is a group of genes that work together and are controlled by one promoter. Think of it like a team sharing a single manager. The lac operon is a classic example. It allows bacteria to break down lactose, but only when necessary. Why waste energy making lactose-digesting enzymes if there's glucose around? When glucose is present, the operon stays off. When glucose is gone and lactose appears, the operon turns on. This smart control saves energy. Me Quiz 2, Operons and Regulation. Q1, what is an operon? A group of related genes controlled by a single promoter. Q2, when is the lac operon turned on? A, when glucose is low and lactose is available. Q3, what does RNA polymerase need to start transcription? A promoter region. Section 5, Gene prioritization and the lac operon. E. coli prefers glucose because it's easier to digest. Researchers Jacob and Monod discovered the lac operon system in the 1950s. It turns on genes only when lactose is the only available sugar. They learned that. The promoter is like a green light for RNA polymerase. A repressor covers the promoter when glucose is present. When lactose is around, it binds to the repressor, lifting it off like removing a roadblock. Mini Quiz 3, Bacterial sugar preferences. Q1, why do bacteria prefer glucose over lactose? Glucose is easier to digest. Q2, what molecule removes the repressor from the lac operon? Lactose, or allolactose, its isomer. Q3, what happens when RNA polymerase is blocked from the promoter? A, the gene is not transcribed. Part 1 summary. DNA, RNA, protein is the central dogma. Transcription and translation happen back-to-back in bacteria. Operons help bacteria save energy by controlling gene expression. The lac-C operon helps E. coli only use lactose when glucose is gone. Part 2, Genetic Engineering and GMOs. Section 1, what are GMOs, Bara? Genetically modified organisms. GMOs are organisms whose genes have been changed by humans. Examples include glowfish, glowing gene from cuttlefish bacteria inserted into zebrafish. Flavor saver tomatoes, engineered to ripen slowly. Fast-growing salmon, grows twice as fast due to modified growth hormone genes. Eco-pigs, pigs that burp less to reduce methane emissions. Section 2, Natural genetic transfer. In nature, bacteria already transfer DNA. Plasmids, small circular DNA used by bacteria. Crown gall tumors, caused by bacteria inserting DNA into plants. Scientists copied this idea to develop genetic engineering tools. They also used viruses and the lysogenic cycle to sneak genes into host cells. Mini Quiz 4, GMOs and nature. What is a plasmid? A small circular DNA molecule used by bacteria. Q2, what plant disease inspired genetic engineering? Crown gall tumors caused by DNA transfer. What is the lysogenic cycle? A viral process where the virus integrates its DNA into the host's genome. Section 3, gene guns and DNA transfer. Gene gun shoots DNA frozen in microscopic ice into plant cells. Only works well on plants, later replaced by better techniques. More precise method, use plasmids to insert genes into bacteria or other organisms. Section 4, Genentech and human insulin. Genentech, a biotech company, made history. Took the human insulin gene, inserted it into E. coli using a plasmid. Bacteria then produced real human insulin. This replaced pig insulin, which often caused allergic reactions. It was also cheaper and easier to produce. This success led to Genentech becoming a biotech giant. And the first real GMO product was born, human insulin. Mini Quiz 5, Genetic Engineering Breakthroughs. Q1, what was the first commercial GMO product? A, human insulin. Q2, why is human insulin better than pig insulin? Fewer allergic reactions and cheaper to produce. Q3, what company pioneered insulin production using bacteria? Genentech. Part 2 summary. GMOs can grow faster, ripen slower, and even help the environment. Nature-inspired gene transfer techniques using plasmids and viruses. Genentech revolutionized medicine by engineering bacteria to make human insulin. Final review, super quiz. Q1, what molecule reads DNA to make RNA? A, RNA polymerase. Q2, what organelle reads RNA to build proteins? Ribosome. Q3, what sugar do bacteria prefer to digest first? Glucose. Q4, what controls whether an operon is on or off? Repressors and availability of sugars. Q5, what was the first widely used GMO in medicine? Human insulin made by E. coli. Would you like this turned into a clean PDF or audio recording next?